Hello and welcome to today's webinar in Knowledge Miles, the 695th Lord Mayor's Lectures. In this free online lecture series, we're addressing the connections in and around the square mile, what Michael Manelli, the current Lord Mayor, calls the world's coffee house, and how those connections might help us tackle future global challenges. Well, today's lecture is a treat. It's entitled How Air Pollution Causes Lung Cancer in Never Smokers. My name is Richard Harvey. I'm a professor at the University of East Anglia and a former professor at Gresham College in the City of London. I'll be convening this session and moderating the Q&A portion of today's lecture. A small apology about my voice, which is on the way out, but I think it will hold out for this lecture. A bit of housekeeping from me. You should be able to see an agenda slide for today, so you can see what's going on. Charles has got some pretty serious material to discuss, and we should give him some leeway to do that, I think. So the Q&A might be a little bit abbreviated, which is a great opportunity to introduce today's speaker, who is Professor Charles Swanton, FRS, who is the Deputy Clinical Director at the Francis Crick Institute, which, for those of you who don't know, is a £700 million building behind the British Library. Parts of it are open to the public, actually, and worth a visit. Now, Charles' CV is summarised on the Gresham Society website and on the Crick Institute's webpage, but it's long, and distinguished. What I would say is it's pretty unusual nowadays for the general public to hear a tip-top scientist, an FRS, talking about absolutely his latest work. And this work, I think, was front page of Nature in April 2023. So most scientists never get a Nature paper at all, but to get a Nature paper and be front cover, I don't know what that's the equivalent of. Being on the front of Vogue, I suppose. Anyway, so Charles, I'm super grateful for you for doing this, because I know you're very busy. And over to you.
Thank you. Thank you very much indeed. I need to share my screen, but it's stopped me from. Here we go. There we go. Right. So I'm in business. Please tell me if you can see it. I hope you can. Can you see that?
Can. Yeah, we can.
Great. Lovely. Right. Thank you so much for the kind intro. It's a huge pleasure to be speaking to you all today. To galvanize interest in this problem, which I hope you'll agree with me, is a problem that we need to reckon with and tackle rather urgently. So we're particularly interested in lung cancer in the lab and this conundrum of how cancer in never smokers begins. Obviously, the overwhelming risk factor for lung cancer is smoking, but I would say there are still about 5,000 new cases of lung cancer in never smokers every year in the United Kingdom, which is almost as many as melanoma and renal cancer combined. So this is an area of unmet need. Now, it's distinct from smoking-associated lung cancer, in that when you sequence these tumor genomes from patients with never smoking lung cancer, they have a very low number of DNA mutations in them. And 50% of patients with this disease have a mutation in a specific receptor called the epidermal growth factor receptor, EGFR, which I will mention numerous times in my talk. And it's also, interestingly, more common in women than men, somewhere between 1.5 and 4 times more frequent in females than males. Potential risk factors for this disease are what you inherit from your parents, so differences in your genome compared to your neighbor or your friend that might determine risk, radon gas, secondhand smoke, and environmental air pollution that I'll be speaking about today. Now, the conundrum here that we've been tackling really and battling with over the last decade is that radon gas, secondhand smoke, and air pollution are all carcinogens, yet there are no carcinogen-induced mutations in these cancer genomes. Remember I said at the beginning, these are genomes that have very low numbers of mutations in them. So what is happening, and how could these carcinogens cause cancer if they're not mutating DNA? Now, this is just to sort of elaborate on this point about mutations again, because it's fairly central to this talk. When we sequence a patient's tumor genome, there are 4 billion nucleotides or base pairs that make up that cancer. Now, in a smoker, a patient who's been smoking, let's say, 1 packet a day for 40 years, they would have what's called a 40-pack year history of smoking. And that's sort of the average number of cigarettes a patient has smoked in our study called TRACER-X. And this is a longitudinal program following patients up with lung cancer who've had their disease resected with the intent of cure. About 50% will relapse and die of their disease. And the study is aiming to understand why that is. So what we do is we sequence their tumor genomes at diagnosis, and we plot on this graph here the number of mutations in their tumor genomes. And on the x-axis, you can see whether there are smokers or never smokers. And the never smokers are all in blue, and the smokers are all in grey. And what you can see is the smokers are on the left-hand side of the plot with many more mutations in their tumor genomes. And we estimate that ever smokers, so those who've smoked at some point in their lives, have approximately 100 particular base pair mutations, so-called C to A tumor mutations per pack year smoked. So in a typical patient from TRACER-X, that'd be about 4,000 C to A mutations in their tumor genomes. Now, the never smokers, obviously are on the right-hand side of this plot, have got a very, very low number of mutations in their tumor genomes. They're shown here. So we've got a conundrum, as I mentioned. Pollution, if it causes lung cancer, is a carcinogen. And if it's a carcinogen, why is it not mutating the DNA of tumor genomes? What is happening, and how could these carcinogens cause cancer without mutating DNA? Now, this is just to remind you that the association of air pollution with lung cancer is not a new phenomenon. Doll and Hill first put forward this possible association in 1950, and it's been substantiated by many groups across the planet, showing a very close association between air pollution and risk of lung cancer. Now, I would argue that if Doll and Hill had studied air pollution rather than tobacco-associated lung cancer, I think our understanding of cancer in general will be very different from how it is today, because it turns out that tobacco smoke is probably an outlier in the context of how a carcinogen causes cancer. So about 7 or 8 years ago, when we set out on this path and this project, we reasoned that if environmental air pollution was going to was a cause of lung cancer in never smokers, it would have to fulfill 4 criteria. Firstly, it would have to explain the geographical distribution of the disease. Secondly, it would have to prove that pollution directly causes lung cancer, and if so, how it could cause cancer without directly causing DNA mutations. And finally, we'd have to explain why cancer in never smokers is more common in females, something that I won't have time to talk about today, but we could discuss at the Q&A. So if you'd asked me 5 years ago, Charlie, how does cancer start? I'd say we pretty much had sewn it up and we pretty much understood how cancer starts. And this is sort of summary of work over 10 or 15 years from the community. Essentially, the classic mutation model. A carcinogen mutates DNA. That confers a selective advantage to one of these cells that then grows out and ultimately forms a tumor. But there's a big problem with this model that we and others had ignored, I think it's fair to say. The first is that the commonest mutation in melanoma, skin cancer, that you all know is due to UV light caused by ultraviolet light, the sun rays causing sunburn that causes cancer. We know that sunlight causes specific mutations, C to T mutations in the tumor genome. And yet this common mutation in melanoma that occurs in about 50% of patients in their cancers, the so-called BRAF V600E mutation, is not a UV light-induced mutation. So if the commonest mutation in skin cancer is not caused by UV light, when I explain to patients why they've got cancer, that it's due to UV light causing mutations, that's clearly wrong. Secondly, normal healthy tissue, it turns out, is essentially a patchwork of mutant clones, many with mutations that are often found in cancer. So this tells us that cancer mutations may be necessary for cancer induction and initiation, but they are by no means sufficient, because if it were, we wouldn't have a normal eyelid. Anywhere you look in the human body, whether it's in the gut epithelium, gut skin, or our own skin exposed to UV light, as you get older, you become a patchwork of mutant clones, and yet eyelid cancer is incredibly rare. And the most, I think, important publication that came out, in my view, in cancer during lockdown in 2020 was this paper by Reaver and Balmain and colleagues, who showed that actually the majority of human carcinogens tested in mice drive and initiate cancer without causing DNA mutations. And really, this was an extraordinary piece of work, because the vast majority of carcinogen assays are essentially based on whether new chemical matter induces mutations in DNA or not. Do these carcinogens essentially drive cancer by disrupting DNA and driving genome instability? And what Alan Balmain showed in this paper is that the majority of carcinogens actually cause cancer by not inducing genome instability and DNA mutations, which raises some serious questions about the introduction of chemical matter into the human environment and whether we really understand how cancers are initiated. So this then mandates we need a second model for understanding how tumours might form. And it turns out this model that's proved so helpful to our work was first introduced in 1947 by Isaac Berenblum, before we even knew what DNA was, before we had DNA sequencers. Isaac Berenblum was the head of the cancer center at the Weizmann Institute in Israel, and he postulated that there were two steps required to form a cancer. The first step is the initiating step, where you need a background elaborated by Alan Balmain and colleagues in the last 20 years. We now know that's the requirement for the initiating step is cells in our normal tissues that harbor mutations that might go on to help a cancer being formed. And the second step is a promoter step. This is an inflammatory step that allows these pre-existing mutant clones in normal tissue to expand and form cancer. Now, the important thing to say about this two-step model is that neither step alone is sufficient to induce a cancer. In other words, the mutation alone cannot initiate a cancer, and the inflammation, the promoter alone, cannot form a cancer. You need both. And so we wondered whether air pollution, because we know it's not causing mutations, might act as a promoter and drive inflammation and the expansion of pre-existing clones present in normal tissue. So with that introduction, I want to show you 3 lines of evidence that supports that hypothesis. First of all, we're going to look at the geographic relationship between this disease, specifically lung cancer in never smokers with this particular mutation I mentioned, EGFR, and the association with air pollution levels, specifically these 2.5 micron particulates that come from brake dust, car exhaust, internal combustion engines, essentially modern living. Secondly, we're going to look to see if these particulates, PM2.5s, promote tumours in mouse models with pre-existing mutant clones. And thirdly, if Berenblum is correct, we would have to show that these pre-existing mutant clones live in or reside in normal tissue as we age. So first of all, let's look at the geographic distribution of this disease. Well, we know, interestingly, that air pollution is more common around the equator. Why is that? It's probably due to the fact that that's where the coal-fired power stations are in southern China and India. But there's also pollution in the United Kingdom, as you know. This is a map on the left-hand side of air pollution levels across England and Wales. And you can see it's centered around the urban environments, particularly London. I hope you can appreciate the M25 here, Heathrow Airport just within the M25, Gatwick Airport just outside the M25, and the Francis Crick Institute right in the middle. Now, what we know from Public Health England data, we accumulated all the data associated with EGFR mutant lung cancer and looked to see if there was a correlation between rising PM2.5 levels across the country and rising incidence of EGFR mutant lung cancer disease. And indeed there was, equating to about 1 extra case of lung cancer per 1 microgram per meter cube rise in PM2.5s across England. So this is a low risk, and the important thing to say is if you were a smoker, the association of risk with smoking is very much higher, about 30-fold increased risk. Here we're talking about much more modest increases in risk, perhaps 5% increase in risk per 1 to 5 micrograms per meter cubed of PM2.5s you're exposed to. Because I haven't got much time, I can refer you to the paper. We saw similar associations at high levels of PM2.5s in South Korea and Taiwan, again with similar risks. About for every 1 microgram per meter cube rise, we see about 1 extra case of air pollution of EGFR mutant lung cancer. So we now need to prove causation, and to do that we have to rely on some specific and rather complex mouse models that I haven't got time to discuss with you today. Suffice to say, what we can do in these mouse models is induce a specific mutation in the lung epithelium, in the lungs of mice, by genetic techniques. And then we can expose mice to air pollution to see if you can enhance tumor formation. And we do this like this. We induce the mutation at day 0, and then we treat mice with air pollutants into the trachea, mimicking high levels of air pollution exposure in places like Beijing, from day 3 onwards for 3 weeks. And then we harvest the mouse lungs at week 10, sacrifice the mice, obviously, and then we count tumors. And we try to do this as ethically as possible and restrict the numbers of mice used as much as possible. And for each of our mouse models, we show a dose-dependent increase in the number of EGFR or KRAS mutant lung cancers that are formed in these mouse models. So as the doses of PM2.5s rise in red, so do the number of lesions formed in these mice increase. So the big question was, is pollution enhancing cancer formation by mutating DNA, classic mutation model number 1, or is it driving cancer formation by the Berenblum model in an initiator-promoter model without driving mutations? And to cut a long story short, 5 years of work showed that air pollution is not inducing more mutations in mouse tumors. And where we see mutations, they have the same signature context. In other words, they're associated with just the natural consequences of aging rather than environmental carcinogen signatures like you would see with tobacco smoke. So air pollution, we think, is enhancing lung cancer formation through other means other than mutating DNA. So how is it working? So to understand that, we have to go back to the drawing board and look at the literature and understand what is the stem cell, the cancer stem cell that ultimately leads to cancer initiation. Well, it turns out this is called the alveolar type 2 cell, the AT2 cell that you'll hear me talk a lot about. Now, to give you an idea, there are 200 billion cells in the adult lung, but only a small minority, about 0.2%, have stem cell progenitor potential. And only a minority of that 0.2% are thought to be the progenitor cell that actually initiates lung cancer. Now, under normal circumstances, this AT2 cell is responsive to this cytokine, which is a chemical mediator from macrophages, a white blood cell, IL-1 beta, which I'll talk a lot about in the next 10 minutes, that signals to alveolar type 2 cells to proliferate, to heal the alveolar lining where gas exchange occurs. So if you are exposed to, let's say, hot chili oil when you're cooking a dumpling or some Chinese food, let's say in the kitchen, or let's say a steak with chili on top, the levels of air pollution rise very rapidly in the kitchen. In fact, I was cooking a steak this weekend, and the levels rose to what would be compatible with a bad day in Beijing. Now that, in combination with the hot oils and the chili, I would contend acts as an irritant that would cause a wound healing reaction in the lung, and for these alveolar type 2 cells to be mobilized and wound healing to occur and gas exchange to be made more proficient after the damage due to the hot cooking oils. But these AT2 cells also are the progenitor cell for lung adenocarcinoma. And so we wanted to see whether these AT2 cells were mobilized after air pollution exposure. And to do this, we rely on a specific technique called RNA-seq, which quantifies the genes that are turned on following air pollution exposure. And what we see are 2 gene programs. One is this alveolar type 2 cell state shown here, but the other is this macrophage program, which I'll come back to in a minute. So we wanted to know if this stem cell wound healing program was being switched on by air pollution. And to do this, we developed with Ilaria Melanchi at the Crick this organoid assay, which gives us a readout as to whether or not there is stem cell progenitor activity following air pollution exposure. And organoid forming efficiency shown here on the y-axis. And what you can see is it's only when these cells from the mouse lung are exposed to air pollution in red and have the epidermal growth factor receptor mutation that organoids can efficiently form. So this really tells us that perhaps Berenblum was right. You need both the mutation and the inflammatory insult for the cancer stem cell to arise. So the question is it the AT2 cell in the mouse itself that's responsible for tumor formation? And we've got very elegant techniques to specifically switch on the EGFR mutation, this activating mutation that contributes to lung cancer, but is not enough on its own to do so. In this specific small population of cells that I said accounts for less than 0.2% of cells in the adult lung. And so we can do this by giving a virus that activates the mutation just in these cells and ask, with pollution, do you see more tumors? And the answer is you do. So in other words, when you just activate this mutation just in this very rare stem cell subtype in lungs and you expose them to air pollution, you get cancers forming. So how does this all happen? How does this all happen? How do PM2.5s promote EGFR mutant cancer from AT2 cells? Well, the first thing we found is we repeated all of these experiments, but in this time, we did it in a particular type of mouse that lacks an immune system. And what we found is, surprisingly, that we see no tumors form, no increase in the number of tumors form when you expose to air pollution, telling us you need the body's active immune system to form cancers in response to air pollution. So we wonder why that might be. And what we saw is this macrophage, a specific white cell gene expression program, pop up after air pollution exposure of these mice. And we wondered if the macrophages might be contributing. We found that the macrophages come into the lungs very abruptly after air pollution exposure. These macrophages are shown in cyan here. These are CD68 positive macrophages that are surrounding the emerging tumors, shown in red. So these light blue macrophages come into the lung following air pollution exposure, and we think trigger the initiation of the cancer. We see the same thing in our lung cancer patients in TRACERx. We see air pollution being associated with more of these macrophages in normal lung tissue. So in other words, individuals who live in areas of the country where there's more air pollution have more macrophages in their lungs. So what must be happening? Well, this is where we turn to a study called the Air Pollution Copper Study, run by Christopher Carlston in University of British Columbia, where he exposed normal human individuals to air pollution or room air to work out what genes are being expressed. And this enables us to look at the similarities between mouse and human to ask what genes overlap between mouse and human. And you'll remember me mentioning this important signaling molecule, interleukin-1 beta. Following air pollution exposure, this is one of the most upregulated genes that's switched on in cells, in mouse, as well as human. So we latched onto this as being a gene, a communicating protein between cells of some considerable importance, particularly as we knew that these AT2 cells that I mentioned need IL-1 beta to adapt to that stem cell state that I mentioned. Where does the IL-1 beta come from? Well, it comes from the macrophages. You can see here, the macrophages are shown in red, and the interleukin-1 beta is shown in yellow. And you can see these yellow dots in the middle of these macrophages, primed following pollution exposure to release IL-1 beta into the alveoli to help with wound healing. Now, why am I so interested in IL-1 beta? The reason is, in 2017, this trial was published in the most prestigious medical journal called the New England Journal of Medicine, which showed that 3 different doses of an anti-IL-1 beta antibody in a trial to limit the risk of cardiovascular disease in high-risk individuals showed a dose-dependent reduction in new lung cancer primaries being formed. Now, this is an important study because the Food and Drug Association in the United States insisted that the CANTOS team collect primary lung cancer outcome data because they're actually worried that more lung cancers would form. In fact, the anti-IL-1 beta did the opposite, and it's really one of the first trials ever that showed a dose-dependent reduction in new lung cancer primaries occurring over a 5-year period in these individuals. So we know IL-1 beta can block lung cancer in humans. Can it do the same in mice? Well, it turns out it can. It can reduce pollution-induced lung cancer from forming in our mouse models. So we know that IL-1 beta is important. We know that it's important and it's released due to air pollution, and the combination of air pollution or IL-1 beta and the EGFR mutation can form cancers. So Berenblum's model seems correct. The question, though, is if Berenblum's model is correct, healthy 50-year-olds like me, and hopefully you, would have mutations in their normal tissue that you would have thought should cause cancer, but will just sit there throughout the lifetime of the individual. So we set about to look for these mutations, because if Berenblum is correct, tumor promoters must act on these pre-existing mutations in these latent pre-initiated cells. So we've got 3 normal tissue cohorts that we can look at, and we find, through very sensitive sequencing approaches, epidermal growth factor receptor mutations present in 15% of normal lung samples. So that's a large number. We estimate that these epidermal growth factor receptor mutations are present in about 1 in 600,000 normal cells from a healthy lung of a 60-year-old. So what happens to these mutations in the presence of air pollution? Well, it turns out they expand. So on this graph here, you can see that in normal tissue from those who've lived in urban environments, these EGFR mutations are more readily detectable and are present at higher frequencies. That tells us that the exposure of the human lung to air pollution allows these pre-existing mutant clones to begin to expand and allow presumably the substrate upon which cancer evolution and cancer initiation occurs to expand. So where do we get these mutations from? Well, the answer is we get them by aging. So this is looking at never smokers. As we age, you see the inexorable increase in the number of mutations that occurs in normal tissue. This is looking specifically at never smokers. So really, I'm afraid the acquisition of these cancer mutations in normal tissue comes about by being exposed to air and water. There's really nothing we can do about this. So what we have to do is try to prevent the inflammatory mediator, reduce our risk globally by reducing air pollution. And this is what we're excited about. So I think Berenblum was right 70 years ago. With the absence of any technological innovation, through his elegant work, he showed that cancer is initiated through a 2-step model. But up until now, we lacked any evidence of what that second hit might be. And I think air pollution is one of these second hits, and there are going to be many others, I suspect, that will act on these latent, pre-existing mutant clones shown here in blue that will just sit there throughout the life of the patient if they're not exposed to the inflammatory mediator. But if you're exposed to air pollution and potentially other environmental insults, this may allow the expansion of these mutant clones to occur and for cancer to ensue. So just rewinding in my last slide now, remember I said at the beginning that the majority of carcinogens do not induce mutations. So this means that they're probably acting through this pathway of driving inflammation and expanding pre-existing mutant clones. But the problem is we don't have assays that can score for whether or not new chemical matter is safe or not, because we just don't understand this process across other tissues outside the lung. So we need these promoter assays to infer the carcinogenic potential of new chemical matter that's being introduced into the environment. Just because it doesn't damage DNA doesn't mean that it's safe. So indeed, how safe is your Tesco's floor cleaning fluid, your furniture polish, or your window cleaning fluid? We simply don't know because we don't have the assays to be able to score. Take microplastics, for example. I read an article this morning on Apple News. There are 200,000 microplastic particles in a 330 ml plastic bottle of mineral water. We've got emerging evidence these microplastics, which we inhale and ingest about 5 grams a week, can do a very similar thing to air pollution in our mouse models. So what other air pollution-like carcinogens out there? Let's think about obesity, alcohol, unknown infectious agents, chronic inflammation, and dietary factors. So we desperately need to establish these inflammatory axes across other tissues. We desperately need to find common inflammatory mediators and pathways that we can inhibit and ultimately attenuate these environmental inflammatory exposures to prevent lung cancer and other cancers from forming in a new era, if you like, of molecular cancer prevention. So much as we give statins to prevent cardiovascular disease, I'm looking forward to a future when we can give a tablet to prevent cancers from being initiated. Thank you very much indeed for listening.
Wow, I mean, that was a wild ride, Charles. I mean, I felt we were tearing through an incredible scientific journey there. So thank you very much for that. Now, as
Pleasure.
predicted, lots of questions. Some of them picking up your own sort of questions, if you like. Okay, well, Tim Connell and Hugh Purser have picked up on something that I think you alluded to, but let's sort of just try and pick it apart a bit, which is in a lot of your work, you're using PM 2.5 as a sort of proxy for particulate
Yeah.
matter. But clearly there are different sorts of particles, you know, tobacco fires and so on. So
Yeah.
there are really two questions there. Really, one is you're obviously content with that as a proxy. What about smaller and larger particles, and what about
Yeah.
particles of different chemistries?
Very good question. So we are very concerned, as you rightly say. This is an issue. So let's start at the beginning. Even within PM 2.5s, there's a heterogeneous mix of particles and molecules and compounds that form 2.5 micron particulates. And the composition of PM 2.5s varies geographically, and it varies by the day and by the season. We took PM 2.5s from a standard laboratory so that people can replicate our experiments in the US. Now we're developing surrogate assays in the laboratory with a macrophage cell line to be able to look for I don't know why it's doing that. I don't know why that thumbs up came up. A macrophage cell line where we can expose macrophages to different particulates, different sizes and different compositions. And we're finding that smaller particles and larger particles also have a similar impact on interleukin-1 beta release. We will be exposing our mice to larger and smaller particulate matter as well to test their cancer-promoting activity. So I think this is a very, very important question. We're doing these studies right now. That preliminary data suggests that smaller and larger particles are equally capable of inducing the inflammatory response. Quite what it is within these particles, we don't know. One of the theories we have is it's LPS, which is bacterial cell wall. Would you believe it, that binds electrostatically to these particles, gets into the lung, deep into the lung. Macrophages in these white cells engulf the particulate matter, and because many of them are inorganic particulates, the macrophages can't digest them, so they sit there in the cell of the white cell, and they can't be spewed out and they can't be digested. And because they contain, or they're enveloped through electrostatic forces by bacterial cell wall, the macrophage thinks, oh, wow, I've got this huge bacteria in my cell. I need to do something about it. And, I'm sorry to think anthropomorphically, but releases these communication molecules like IL-1 beta, TNF, IL-6 that signal to other immune cells to come in and say, look, we've got a problem here. We've got a bacterial infection. So that's one of the theories we think may be happening here, that this is sort of they're signaling as if they're bacteria.
God, what a complicated world you live in. I mean, it's amazing you can think your way through this. So Peter Cousins asked about ILB. It was IL-1B, wasn't it?
IL-1B, yeah.
Yeah. He says, do you think it will be a universal mediator, or will the mediator vary according to
Wow,
tumor type?
that is a great question. Funnily enough, we had a meeting yesterday about exactly that with an undisclosed pharma company, because we're very keen on a sort of prevention strategy. And the answer to your question is the preclinical data, similar to what we've shown you in mice, has got there's pretty strong evidence for IL-1 beta being important in pancreas cancer through similar mechanisms. Mesothelioma, which is a cancer of the lining of the lung that's associated with asbestos, possibly gastric cancer, possibly colorectal cancer, possibly subtype of breast cancer. So it may be that IL-1 beta is more important across other tissues as well. But to your point, this is exactly what we're working on now. We're trying to establish common inflammatory pathways where we can, hopefully, the holy grail will be to identify a sort of universal cancer preventative medicine that would dampen down inflammation. Now, the problem, of course, is probably one of you is going to ask me is, are there side effects of reducing inflammation? And in the CANTOS trial, where patients were treated with anti-IL-1 beta, there were more deaths in the treatment arm due to pneumonia and sepsis, because you're dampening the macrophages. They can no longer do their job of spotting real bacteria, so to speak. And so any medicine that we're going to use to prevent cancer obviously would have to balance the benefits against the risks, and we'd have to come up with ways of boosting the immune system to spot bacterial or viral infection, so that inflammation occurs when it needs to, not when it doesn't.
Yeah, it's such a tricky. Yeah,
It is.
fiddling with
It
the
really
control
is.
loop.
Yeah.
Fiddling with the control loop is always very tricky,
Yeah.
isn't it?
That's right. Yeah.
So Frank Moxon said, "Look, there seem to be some non-smoking outliers in your charts. Presumably, they were
Yeah.
exposed to high levels of contaminants. I think I
Yeah.
noticed the poor people of Southeast London were an outlier at one
Yeah.
point in one of the
No, I
charts."
mean, so if I'd had more time, I'd have elaborated. Epidemiology is an extremely complex science. I thought animal work was hard, but epidemiology is extremely hard. The problem with epidemiology is you cannot control for every variable. I would have shown you the data that I've shown you is very specific for a subtype of never smoking lung cancer, the EGFR mutant variant. We've got data from UK Biobank, which looks at all lung cancers and shows a very strong correlation. But also there are data from 5 or 6 meta-analyses with 500,000 patients, 300,000 life years, shows a very statistically significant correlation between air pollution and lung cancer risk. Part of the reason for these outliers is because the particular data I've shown you is only a very minor contribution to the literature. It's really to sort of introduce you how we got into this field. It's not by any means robust epidemiological data. It's just to justify why we use the type of mouse model we use. And as you rightly put it, there are outliers, and that's in part due to the fact this is an ecological analysis. We didn't have postcode specific data for every patient in that Public Health England data. We only had regional level. So it's like saying Southeast London is a large place. Some of those areas will have very high levels of pollution, some won't. So it's a very crude ecological analysis, and not one I'm hugely proud of, but one that convinced us that our mouse models would be worthy as a model to start using. But I could show you, a lot of meta-analyses data that would convince you that air pollution and lung cancer are tightly associated.
So, Charles, we've got a few minutes left, but I wanted to, well, there are too many questions to handle as usual, but women, your final question was about women, and Charlotte Dorber Ashley has asked that question. It really was a head scratcher, because when I looked at your scattergram of by age of how likely it was that you were to get various mutations, women didn't look to me like particularly more likely than men. But I think you do have an answer to it. You just eliminated it.
Yeah, can I share my screen? Can I share my screen? I've got another slide I'd like to just show you very quickly, if that's all right.
I'm sure that's possible.
Just to break down the data by. Okay, great question. Thank you for asking it. This is the data I've just shown you for Public Health England, broken down between females and males. You can see females are about 50% more likely to get lung cancer than men. Now, we don't know why that is, and that's in the UK. In Singapore and parts of Southeast Asia, it's probably 2, 3, even 4 times more common in women than men, to the extent where, I mean, it's an extraordinary statistic, this, but 60% of new lung cancer diagnoses in Singapore, I'm led to believe, are in women, female non-smokers, whereas here in the UK, it's probably 5%. So it's a very, very common disease in parts of Southeast Asia. So the question is why? And so the grant that we've got in submission with the European Research Council, that I do hope they'll fund, is looking at 3 potential reasons. One is behavioural differences. I mean, it could just be, I'm afraid to say, a reflection of the patriarchy. More women than men cook. And actually, I think this is our most likely scenario. I mean, I told you about the data from my kitchen last weekend, 100 micrograms per meter cube. That's 4 times higher than anywhere in London. And it's high for a long time, even with an extractor fan running for 4 hours. By the end of 4 hours, it was still 60 micrograms per meter cubed. So if you think about people exposed to hot cooking oil, I think that's an issue. Then there's Differences between males and females. Women have two X chromosomes, and there are genes on the X chromosome that are completely inactivated. And then there's hormonal reasons. The lung cancers express the estrogen receptor, so there may be differences in biology between women and men. This is just to show that the two cell types I mentioned, the macrophages and these 82 stem cells, they interact more in females than males. And so it might also be that female lung is just more inflammatory than male lung. That's another reason. And there may be biological reasons for that in the context of pregnancy, maternity, and what have you.
Wow, that's so interesting. I mean, we're out of time. We're going to have to stop taking questions. There are too many questions.
Cooking oil is my number one. Cooking is my number one reason.
I mean, what a detective story. You know, I mean, I would have given up right at the beginning. It's too complicated.
Well, we nearly did many times, but there were a few things that made it possible.
Huge kudos from us for sort of just cracking on with it, and it's an amazing story. Now, just a quick look forward, folks. We have some lectures coming up in this series, and they're really fascinating. Wreck of the Gloucester I know something about, London Science City, and so on and so on. If you'd like to know more, please do have a look at the Gresham Society website. Gresham Society is a sort of supporters club for Gresham College. If you like this sort of thing, then you'll probably enjoy lectures at Gresham College, so do have a look at their website. So, two bunches of thanks. Firstly, thanks to everyone who attended and all of your fantastic questions, which were I really enjoyed and really made me think, so that's super. But most of all, thanks to Charles. I know he's super busy. He's a very distinguished scientist. It's probably the last thing he wants to spend his morning doing.
I really enjoyed it. Thank you.
Really good. And I really felt we were on the sort of edge of the bleeding edge of science, which is very, very enjoyable for me. This lecture will be available on YouTube in case you want to revisit it. Other than that, thank you very much and goodbye.